Welcome to this video on the structure and function of genetic material, presented by Olorontoyan, JT. Genetic material is the biological blueprint that stores, transmits, and expresses hereditary information. In humans and most organisms, this is DNA. DNA stands for deoxyribose nucleic acid. The understanding of DNA as hereditary material evolved through key experiments. Griffith's experiment in 1928, followed by Avery, McLeod, and McCarty in 1944. And finally, Hershey and Chase in 1952 confirmed that DNA carries genetic information. The structure of DNA can be examined by looking at its chemical composition and its double helix structure. DNA is a polymer of nucleotides. Each nucleotide consists of a deoxyribose sugar, a phosphate group, and a nitrogenous base. Bases are classified as purines, which have a two-ring structure, or pyrimidines, which have a one-ring structure. This slide visually differentiates between the structure of purines and pyrimidines. In the chemical composition of DNA, the phosphate and sugar molecules form the backbone of the strand, while the nitrogenous bases project inward. The DNA double helix model, designed by Watson and Crick in 1953, consists of two antiparallel strands. Adenine pairs with thymine and guanine pairs with cytosine. This is known as complementary base pairing. The strands twist around a common axis, forming a right-handed double helix. The structure is stabilized by hydrogen bonds between the bases and by base stacking. There are three main types of DNA: aDNA, bDNA, and zDNA. aDNA is a right-handed helix, shorter and more compact than bDNA. It is rare in living cells and is observed in DNA-RNA hybrids and double-stranded RNA molecules. bDNA is the predominant form in human cells and the classic Watson-Crick model. It is a right-handed, long, and thin helix responsible for storing and transmitting genetic information. zDNA is a left-handed helix with a slim, elongated zigzag backbone. It typically forms under high salt concentrations and plays a regulatory role in gene expression and genomic stability. The main functions of DNA include the storage of genetic information, replication, mutation and variation, and acting as a template for transcription and gene expression. DNA stores hereditary information in the sequence of its nitrogenous bases, A, T, G, and C. Each unique sequence forms a gene containing instructions for synthesizing specific molecules. DNA replication is the process of making an exact copy of itself before cell division. This ensures genetic continuity and preserves the same genetic information in every cell. During replication, the two DNA strands unwind, creating a replication fork. Each strand serves as a template. Enzymes like primase and DNA polymerase help add new complementary nucleotides in the 5' to 3' direction. A DNA mutation is a permanent alteration in the DNA sequence caused by replication errors or environmental damage. Mutations are essential for evolution, but they can also lead to diseases. Types of mutations include point mutations, chromosomal mutations, and induced mutations. Point mutations result from a single base change. These include substitution, insertion, or deletion of a single base. Chromosomal mutations involve large-scale changes in chromosome structure, such as duplications, deletions, inversions, or translocations. Induced mutations are caused by external factors called mutagens, like UV radiation, chemicals, or infectious agents. DNA acts as the template for RNA synthesis, initiating gene expression through the central dogma of molecular biology: DNA to RNA via transcription, and RNA to protein via translation. Transcription copies a gene's DNA sequence into mRNA. Translation occurs when the mRNA binds to a ribosome, and its codons are translated into amino acids to form a protein. Amino acids are the building blocks of proteins, and their sequence is dictated by codons, which are triplets of nucleotides in the mRNA. Codons include start, sense, and stop codons. This diagram visually represents the central dogma of molecular biology, showing the flow of genetic information from DNA to RNA to protein. For further study, please consider differentiating between A, B, and zDNA, discussing the functions of DNA, and explaining the structure of DNA with a diagram.